which is known as ATPase, and this ATPase has two subparts. One is known as, this is the part which is in the membrane. It is known as a proton transporter. It will allow the proton to enter. And then the other part, this part is the one which is responsible for the, for synthesis of ATP. So this part is basically responsible for synthesis of ATP. So this way, your energy is being produced. To give you some more details, we have already talked about what is a mitochondria. You have a cristae, you have inner mitochondrial membrane, and inside this you have a matrix. This just gives you an idea where the citric acid cycle occurs, and citric acid cycle is close to the mitochondrial membrane. Why it is important to know where citric acid cycle is happening is because it is the citric acid cycle where your NADH and FADH2 are being produced, and they are the one which are the donor of electron in electron transport chain. So in oxidative phosphorylation, NADH and FADH2 are used to reduce molecular oxygen. So it doesn't happen in one step. It is a several step process. So that is what we will be talking about. And the high exergonic reduction of molecular oxygen by NADH and FADH2 occurs in a number of electron transfer reactions. So they will be individually talking about these electron transfer reactions, which are taking place in a set of membrane protein known as electron transport chain. So electron transport chain is defined. So here you will not find that one protein is involved. There are several proteins are involved. So first we will talk about the complexes. The complex which is transferring the electron from one part to another part, but they are held together, and that's why we call it the complex. So in this oxidative phosphorylation, electron transfer potential of NADH or FADH2 is converted into phosphoryl group transfer potential. So it's very clear. You have learned what happens in ATP. ATP has a phosphoryl group, terminal phosphoryl group, which when transferred, it provides energy. So you know that delta G is minus 7.3 kilocalorie per mole when ATP is transferring its phosphoryl group. So the measure of phosphoryl transfer potential is known to you. It is delta G0 prime. And similar kind of mechanism happens. So what happens when delta G0 prime, we say it is for the hydrolysis of activated phosphate compound. And that's the way you give the phosphoryl group transfer potential. The corresponding expression also exists for electron transfer potential. In fact, you might have learned this in your electrochemistry while in class 12th and in subsequent classes, where the electron transfer potential can be given by E0 prime and is known as a reduction potential. They can be also called as a redox potential or oxidation reduction potential. And what you require here is that you require the oxidized form of X, any form, and then you need a reduced form to X minus. So such a pair are called as a redox couple. The reduction potential of this couple can be determined by measuring the electron motive force, and this can be generated by sample half cell. So we are referring to the same concept which you have learned in electrochemistry. You have a half cell connected with the standard reference half cell, and you know that standard reference half cell are made up of proton and H2 couple. So that is what we are going to just reiterate. So if I take a standard reference half cell, which consists of an electrode immersed in 1 molar proton solution that is in equilibrium with hydrogen gas at 1 atmospheric temperature. So this is a standard we use across the electrochemistry. And if the reaction proceeds in the direction you have X minus plus proton, it gives you X plus half H2. So this reaction is called as reaction in half cell or half reaction or couples. And then you should have another half of the reaction. So here you have X minus, which gives you X plus electron. And on other words, you can say proton plus electron gives you half H2. So why I'm discussing is because it is very important that we should know that when one electron is being transferred from NADH to molecular oxygen, how much energy we are going to generate. And so that means if I want, I can convert this reduction potential into delta G value. So you have a interconversion, and this equation is known to you. It is delta G0 prime is given by minus NF delta E. So looking at this reaction, let us see what is the redox potential when your pyruvate is converted into lactate. So you have seen that pyruvate to lactate conversion is what? Is basically regenerating NAD+. So one can uh, divide the same reaction into two halves, and each half we can measure this electrode potential, and this potential can be finally found out how much energy can be generated by a couple or when the electrons are being transferred. This is the one which gives you a very simple oxidative phosphorylation measurement how you can do for oxidation reduction potential. It's a simple technique. You might have done it, or at least you have been taught about this in your electrochemistry. So one of half is the standard one, and here you keep your redox couple. So from the redox couple, you can measure the changes, whatever is occurring when the electron is, and this bridge is given for continuity of the electrons flow. This is not to be remembered, but to known that what does it mean. So when I'm talking about NAD plus to NADH plus plus, so you have oxidant on left hand side, and on the right hand side you have reductant. And each one, what you have is that the number of electrons which are being transferred and the redox potential for this. So what you see here is that NAD plus and NADH, they have a redox potential of minus 3.2, minus 0.32, and whereas a molecular oxygen has in the plus. So that means the electron can flow from NADH to molecular oxygen. So this is a very simple oxidation reduction couple, which is showing that how the electrons are flowing from NADH, which is highly, which has very high electron donating potential, to molecular oxygen, which is having high electron accepting potential. Now the concept here has been further discussed, that how this, will there be any rate of electron transfer? How it will depend? So of course it depends on the medium. So electron transport rate will depend on the medium, depending on how much distance your donor and acceptors are for electron, it will happen. It can happen in vacuum, but in case of a protein, what happens that this electron transfer rate decreases by a factor of 10 for every 1.7 angstrom distance increase. What it means is that if the proteins are far apart, the electron transfer rate will be very, very slow. And that is why you will see that in the electron transport chain, there is not one protein, there are several proteins, small proteins, they are associated, and that's what makes the electron transfer chain. And this is again some calculation which shows that if I take half reaction, taking molecular oxygen to water, what is the electron transfer potential? And same way, when NADH is transferring its electron and is becoming NAD, what is the electron transfer potential? And based on this, if I write that the transfer of electron from NADH to molecular oxygen, you can find out what is the delta G contribution for this reaction. And delta G, if I substitute based on whatever the electron transfer potential you calculated, having molecular oxygen and using NADH to NAD plus conversion of or transfer of electrons, what you find is that the delta G value, and these are the values. So what you do is that for first reaction you calculate the value putting NFE, and for the second reaction you put the NFE value. So first I have put delta the value of E0 prime plus 0.82 volt, and in the second case I have put minus 0.32 volt. So these two have been put, and then what you find is that the delta G value for these two coupled reaction is the one which comes to minus 52.6 kilocalorie per mole. What does it mean? That means if I talk about the number of ATP, so it is possible that this energy, free energy, which is minus 52.6 kilocalorie, should be capable to synthesize 3 molecules of ATP. Each molecule of ATP, is having delta G0 value when ATP hydrolysis occurs is minus 7.3 kilocalorie, which is much lower than what you have generated in terms of energy. So delta G0 value which has been generated here is much higher than the amount even required for the ATP synthesis. So what happens, this amount of energy, free energy which you have generated when NADH is giving donating its electron to molecular oxygen. During this flow, the amount of energy which has been generated is highly potential to generate 3 molecules of ATP. And that is what has been considered. Of course, in different cases, different assumptions, number may be different, and in books also you may find different numbers, but at least what you are showing is that theoretically it is possible to synthesize 3 molecules of is sorry, you can synthesize 3 molecules of ATP when such kind of electron transfer occurs. Is it clear to you? So far the concept? Any question? So far we have been discussing about the the concept how electron is going to flow back, flowing from NADH to molecular oxygen. Now we are going to talk about what actually pump are and how they are flowing. So you find actually that this respiratory chain, they consist of 4 complexes. What are these 4 complexes? So you have NADH. NADH will give its electron to the first complex, which is known as NADHQ oxidoreductase. And from here, electron will flow. And this protein, when I'm talking about this is a complex, that means it is not a single protein. The name has been given one, it will be consisting of something like 34 small protein. Mostly they are FMN and iron sulfur clusters. So this part we will discuss further. Then what happens that the electron shuttles from NADHQ oxidoreductase to Q cytochrome C reductase. So this is a coenzyme Q, or the is something which is present, and it can shuttle the electron between NADHQ oxidoreductase to coenzyme Q cytochrome C oxidoreductase. So these are oxidoreductases. These are the enzymes which are present in the subunit structure, and they are the one which are responsible for transferring electron. And then the next step, cytochrome C, which is a soluble protein, water soluble protein, which can transfer electron from cytochrome C oxidoreductase to cytochrome C oxidase. And finally, electron is being transferred from cytochrome C oxidase to molecular oxygen. Now, some of you must be seeing this succinate Q reductase. This is another complex. And can you recall where we have succinate dehydrogenase? Where do we have? Where do we find this enzyme? This enzyme is present in citric acid cycle. So, and if you recall here, one molecule of FAD is converted into FADH2. So the electrons which is present in FADH2 form, which has been extracted from the oxidation of fuel molecule, is going to be present in succinate Q reductase, and where it will be bound in the form of FADH2. And the electron moves from FADH2 to coenzyme Q. Now, what is important here is you see that NADH is going to have high negative value for electron transfer potential than the coenzyme, than the succinate Q reductase, where your FADH2 is linked. So whenever electron flows from NADH to molecular oxygen, the number of ATP produced will be larger than the one which is being produced from FADH2, which is bound to succinate Q reductase. Now let us see this concept. So electrons are transferred from NADH to molecular oxygen through a chain of three large protein complexes. And these complexes I have already described about: NADH-Q oxidoreductase, Q cytochrome C oxidoreductase, and cytochrome oxidase. So these are the three. One is this one, second, and third one. Then electrons are carried from NADH-Q oxidoreductase to the cytochrome C oxidoreductase and second complex of the chain by reduced form of coenzyme Q. So it is same thing which I have described earlier, that this electron is moving through coenzyme Q. Okay, so and which is known as ubiquinone. Why it is known as ubiquinone? Because it is ubiquitous. You will find in the organism starting from the bacteria to human being. That means it is present everywhere, and that's why the name has been given ubiquinone, because it is ubiquitous. Now, cytochrome C is a small soluble protein. That is what I said. This cytochrome C is the one next one, which is a soluble protein, and it will shuttle the electron from cytochrome C oxidoreductase to cytochrome C oxidase. So that is what is interesting. And then NADH-Q oxidoreductase, succinate Q. So you have these as a complex one. So whenever first time when they have isolated these proteins, they were protein in the form of complexes. So you have first complex, which is this is the first complex, red color. This is the second complex, succinate Q reductase. I have not given it color. This is the third complex, and this is the fourth complex. So these are the fourth complexes. In some places, you will see that ATPase has also been given as a fifth complex. So these are the complexes. Complexes doesn't mean anything that other than that if you take mitochondria, you want to solubilize, remove the lipids, you will get these protein complexes. So the reaction catalyzed over here is, before we move on to this, let us talk about the other thing. So you have all these complexes, four complexes. Complex 2, which I said is succinate Q reductase, is in contrast with other complexes, does not pump protons. This part we will discuss a little, in detail. What happens that whenever electron transfer from one of the complexes to another complex, so suppose electron is transferring from this to this, what will happen? So NADH to this, this to this. So once the electron is being transferred, every time from here, it is presumed that one, I'll just show it to you by selecting this. So what happens here? One proton is going to be pumped out. Here, one proton is being pumped out. Here, one proton is being pumped out. And if I say that every time protons are being pumped out, one molecule of ATP is synthesized. So when NADH transfers the electron to molecular oxygen, there are three places where protons are being pumped out. So classically, it is assumed that when electron transfers from NADH to molecular oxygen, 3 ATP is produced. Now tell me, what will happen when electron is transferred from succinate Q reductase bound FADH2? So if electron enters from here, it is skipping this step. And that is why only there are two places where electrons are electrons are moving through these complexes, and two places where protons will be pumped out. And because of that, the number of ATP being produced by the FADH2 will be one less than NADH. So this is a very simple complex which concept, and where electrons are being moving from one complex to another complex, how ATP is being generated. Now the electrons of NADH enters into NADH-Q oxidoreductase. We have already discussed this. It is having 34 polypeptide chains. So what I have said earlier, that this complex is not a single one, though it has been shown. It is a complex of proteins. So there are 34 small proteins. Mostly it is iron sulfur cluster, but yes, to begin with, it also has FMN. So here, the construction of this pump is like other two respiratory chain, is cooperative effort That means the protein which is present here is coded by genes which are present in mitochondria and nucleus. That means it is not dependent on the nucleus completely. So this protein is synthesized with the help of gene which is present in nucleus as well as the mitochondria. It's just, based on how the mitochondrial proteins are being synthesized. Second thing is that the reaction catalyzes uh, conversion of NADH to NAD plus, and here your Q is involved. Q is being converted into QH2, which is quinol, and your protons are being pumped out. The initial step in the binding of NADH and transfer of its two high potential electron to FMN, which is a prosthetic group of complex, to give reduced form of FMNH2. What it is, because it's a relatively complex, because I have not shown you all the reaction here, but as I said, NADH Q reductase will have FMN. It will also have lot of iron sulfur cluster. So don't get confused. Just think of that you have a complex, and this complex is the one which has. So if I say this is a complex, it will have FMN, it will have lot of iron sulfur cluster, and those cluster and the plus iron sulfur cluster, FES, all of them, they will make 34 polypeptide chains. So there are 34 small chains. So it also clears the idea that when electrons are being transferred, the proteins are not far apart. They are very close to each other, and that's why the rate will not be decreased. So this part we have already discussed, that how the presence of the acceptor and the donor will help in the transfer of electrons. So here electrons are moving, and ultimately what will happen, that it will convert Q into QH2, and that is the way the electron transfer happens. Now ubiquinol, the Q which was present as a shuttle, that is converted into QH2. Now this QH2 is the entry point for electrons from FADH2 of flavoprotein. What it means? It means is that this QH2, so suppose the electron has entered, has moved from NADH Q oxidoreductase to Q, which has become QH2. That is the point where the electron can also enter from FADH2. Okay, this is the place where it can enter from succinate Q reductase complex. It can also enter from other cycles, for example, from glycerol phosphate dehydrogenase and fatty acyl CoA dehydrogenase. So this is the point where the electron can enter through FADH2. So wherever FADH2 is there, they will allow the entry to Q to form ubiquinol. So this is the point where you can say that this is the place where electrons are coming from different sources, either NADH or FADH2. FADH2 is present in citric acid cycle. FADH2 can also be present in fatty acyl CoA dehydrogenase. I have not talked about fatty acid synthesis or fatty acid degradation, so I'm just putting it on hold, and you will be learning that FADH2 is being produced during the fatty acid metabolism as well. Now, succinate Q reductase complex and other enzymes that transfer electron from FADH to Q, in contrast to NADH-Q oxidoreductase, do not transport proton. What does it mean? This I have shown you earlier, that here at this place, electron is being pumped, protons are being pumped out. If electron moves through this is going to pump out a proton. This is going to pump out a proton. So all these places, when electron transfer, there is a pumping of the proton. But when electron transfer from here to here, succinate Q reductase, which is the one which is responsible for the binding of FADH2, when electron is transferred from here to coenzyme Q, there is no proton which is being pumped out. That means here, when electron moves from NADH to molecular oxygen, you have higher amount of proton pumped, and that means that you will have more amount of energy or ATP being produced. Okay, so let us move on how the electrons flow from ubiquinol to cytochrome C through coenzyme Q C reductase. Very interesting thing, you will find that the coenzyme Q is two electron carrier, and then it becomes single electron carrier at the later stage. So what happens, one of the electrons is being recycled, and that is where the so that recycling is important, which we will be discussing. So the function of coenzyme Q of cytochrome C oxidoreductase is to catalyze the transfer of electron from QH2 to oxidize cytochrome C. And it will pump proton, as you have learned, is pumping of the proton. Proton is coming out of the mitochondrial matrix. Then what happens that QH2 is converted into Q, and during this process, because QH2 has to get protons, so this proton comes from the matrix, and then again it is pumped out. Otherwise, what will happen in mitochondria? All the protons, they will be lost, and that is why you keep on getting protons from the and the re-entry of the proton, they will also happen. The coenzyme Q cytochrome C oxidoreductase is a dimer with each monomer containing 11 subunits. At this point, it is not important. We are not discussing about the structure. So I'm not interested in giving all the structure. But what is important is that in addition to heme, the enzyme also contains iron sulfur protein, which I have discussed earlier. And this coenzyme Q, C oxidoreductase, it is the one where again you will find it is not a single protein. There are many proteins which are involved, and it shows that how this coenzyme Q, H2, which is quinol, it is pumping out two protons, and then what happens that it is first giving you one electron to Q and another electron to cytochrome, and ultimately these protons are entering in the mitochondria for reducing coenzyme Q, which becomes QH2. And whole process is where QH2 is converted into Q, and again Q will be converted into QH2. It is recycled because these two protons coming from the matrix are going to reduce quinol. So this is a kind of a cycle. Recycling of Q is happening, and during this process, one electron is moving from coenzyme Q to cytochrome C. So this is the reaction which is shown. You have cytochrome C, which is the reduced form. They will get oxidized, and the four protons, they are taken from the matrix, and one molecular oxygen is converted into two water molecule. So the overall process of cytochrome C oxidase. So we have moved on from first complex, second complex, and third complex. So this is the third complex where you are looking at how electrons are moving. So cytochrome C oxidase, the enzyme is cytochrome C oxidase. It is a fourth complex. So which one is the second complex? Can you recall? The first complex is NADH-Q reductase. Second complex is where succinate dehydrogenase is present, which is having a bound form of FADH2. And third one is you have seen where cytochrome. So I'll just give you a kind of understanding. So this is the first complex. Electrons are entering from NADH2. This is the second complex. Electrons are being pumped from FADH2 to coenzyme Q. This is the third complex. So each complex you have learned is having several FMN and iron sulfur cluster. Now we are discussing about cytochrome C oxidase, and that is what is the, you can say, is the last one in the series. So this is the one which can pump third proton and transfer electron to molecular oxygen. So finally, this cytochrome C reductase will be transferring its electron, and what will happen from molecular oxygen, you will get water molecule which is being formed. So here, the best thing is that you learn that molecular oxygen is an ideal terminal electron acceptor. Now, let me tell you that if you recall the oxidation reduction potential, if I have something which has the same redox potential like molecular oxygen, that can also accept the electron from the chain. And but molecular oxygen is very ideal terminal electron acceptor because it has high affinity for electron, and it provides a large thermodynamic driving force. However, there is a problem, and the problem is that if the transfer of single electron occurs to molecular oxygen, what will happen? You get superoxide. And if there are transfer of two electrons, you get peroxide. So that's why you see that these are the reactions which are occurring inside the body, and that could be a problem that could also lead to some kind of a problem inside the body. So this is to recapitulate how the electron moves from NADH to molecular oxygen. If you have any question, I can address that. So this is the last step where it has been shown how cytochrome C oxidase. So this is a complex iron sulfur cluster. It is reduced to cytochrome C is oxidized, and during this process, 4 protons are being pumped out and 4 protons are taken up. So there are the reactions where this electron transport chain is transferring electron and pumping out the proton. So these four protons, they are responsible for converting your oxygen into water, and these 4 protons are being pumped out. So we go back to the same reaction which we started with. If I have NADH and it is converting to NAD plus, we have calculated that delta G is minus 52.6 kilocalories. And that is what you can calculate, that when ATP is being synthesized, there is a plus 7.3 kilocalorie per mole is a value. So if I have 3 molecules, so what will happen? Around 22 kilocalorie is required, and what will happen to the rest? The rest, because the process cannot be 100% coupled, and that is why that will be lost. So ultimately, you can say, suppose this value is less, or this value is just 22 kilocalorie, then this will not be able to synthesize 3 molecules of ATP. So there is some theoretical calculation which has been done. And the question is, what is the mechanism of ATP synthesis? ATP, as I showed you, is consisting of 2 parts. One part is for pumping protons, that is FO, and second part is the F1, which is for the synthesis. So F1 and FO part. This is a complex. Again, this is referred as a complex 5. As I said, some people, they prefer it as a complex 5. So all of them, they are present in the mitochondria. So what will happen? Enter, and when the proton enters, what will happen? There will be, because of the entry of the proton, which is re-entering through the ATPase, that will lead to the synthesis of ATP, and the enzyme involved is ATP synthase. So let us see how ATP synthase occurs, it works. I will agree with you that these are individual subject requires one full class if I discuss in detail, but I want to give you some basic concept. And these concepts are going to be elaborated somewhere in your different courses. So maybe you will be learning about the ATP synthesis if you do bioenergetics course. Suppose you are doing enzymology course, you will be learning all of the enzyme steps. So here, what I'm just focusing on how ATP synthesis is being processed, and ATP synthesis is occurring through enzyme ATPase. And just to give you a basic concept, if I say that ATPase is made up of two parts, not 2 units. So one part is like, so this is the shape which is given generally for ATPase. This is FO. It has been given FO because it is oligomycin sensitive. People, they call it as F0, but I would call it as FO because the nomenclature is based on the sensitivity to a particular inhibitor. And this is known as F1. This is known as F1. Now, F1 is ATP synthase, which is responsible for synthesis of ATP, and FO is involved in the proton pump. So this is the one which acts as a channel for proton. So what you have learned so far? that when electron moves in, so suppose I say that this is a mitochondria, what will happen? That the electron transport will occur here. The electron is moving. Because of the movement of electron, what happens? The protons are being pumped out of mitochondria. Now this proton, which has been pumped out because of the electron transport chain, will re-enter. So how they will re-enter? They will not re-enter from all the places. They will enter through a uh, enzyme, which is ATPase. So ATPase, what you require? You require F0 portion. So there's F0, which is outer part of the mitochondria. F1 is on the inner part. So I'm going to describe about F1, how F1 is working. And this F1, if I recall, there are 3 Nobel laureates who got a Nobel Prize because of the understanding the mechanism, because it's an energy generating mechanism. On the other hand, people, they also call it the smallest molecular motor, which is responsible for doing some job. So what I'm trying to talk about is that this FO and F1 subunit, they are responsible for, F1O is responsible for entry of protons, and F1 is responsible for synthesis of ATP. So what this F1 will do is that it will take ADP and PI, which is already present inside the mitochondria, and it will synthesize ATP. So you get ATP synthesis inside the mitochondria because of the flow of the proton, which is happening through the ATPase. So you know what is the source of energy, and you know that how they can be involved in production of ATP. Now, what is the source of energy? The Mitchell, who got the first Nobel Prize on ATP synthesis, he said, because there is a gradient, there is an electrochemical gradient, and that gradient is responsible for the energy which is helping in the synthesis of ATP. And that's the way he said, he proposed the mechanism, he did a lot of calculation and came to a proof that because of the electrochemical gradient which is being created because of the proton which has flowed outside the mitochondria. So there is a potential difference, and that is why it is responsible for generation of ATP. But then later on, another group which has come up, which says that no, it is not because of the electrochemical potential, it is because of the binding change mechanism. So what he says is that when ATP enters inside the mitochondria, so suppose you have this ATP, and if I say that this is the F1 unit, so F1 unit, I have talked about that this has several subunits. There are alpha unit, beta unit, gamma, delta, all of them, they're present. So what has been proposed by another Nobel laureate, which is known as a binding change mechanism of ATP synthesis, what he says is that when the proton re-enters inside the mitochondria, there is a change in the structure of the protein. And when the protein structure changes, what it does is that it keeps ADP and PI close. So there is a tight binding site, there is a loose binding site. So initially, the ADP and PI bind to the mitochondria, this ATPase subunit, and then they are translocated from one place to another place because of the change in the subunit structure. And the subunit in the T refers to tight form. Here you can see there is a T, and O is open form. So you have them in loose binding form or tight binding form or open form. So in open form, ADP and PI can bind. Then they will be changed to loose form and then to the tight form. And tight form means you are forcing ADP and PI to come close to each other so that they can synthesize ATP. And once ATP is being synthesized, they will move to loose form, and loose form means the ATP will be released from the surface. So again, recycling happens. So what you have learned here in the binding change mechanism, that initially ADP and PI, they will bind to the surface of ATP. This is the synthetic protein which is responsible for synthesis of ATP. then it will change the structure, and when the change in the structure, so you see 120-degree rotation of gamma. So this is a gamma subunit which is responsible for changing the orientation, and when it changes orientation, what will happen? It changes from tight form to loose form, and when it is a loose form, it will be released. On the other hand, the open chain or open form, open part or open subunit is the one where ADP and PI can join, and once they join, the process of changing the structure of protein happens, and during this process it will be converted from open to loose to the tight form, and during this form conversion, your ATP is being synthesized. So this is another mechanism which has been given, and still it is debated even after getting so many Nobel Prize. Still this controversy of how ATP is being synthesized, what could be the source of energy, is not known. But one thing is clear, that ATP synthesis is a link with the electron transport chain. Now you may ask question, how we can prove it? So suppose I block the electron transport. So there are inhibitors. I will show you what are the inhibitors. You can block the transfer of port of electron in the electron transport chain. You see the ATP synthesis is not happening. That's number one. Number two is that if I block the re-entry of the proton, you will see that the electron ATP is not being synthesized. So if you uncouple the electron transport chain and phosphorylation, you see the inhibition. You can also see inhibition if I block the ATP synthase. So there are many mechanisms by stopping the ATP synthesis, you can prove that the electron transport chain is coupled with the ATP synthesis. Is it clear so far?
Yes, it's clear.
Okay, anything, any question, you can come back to me. I will try to answer you all the queries. Otherwise, I'm following completely book from which is Stryer, Biochemistry by Stryer. It is available online. You can read it. It has been given in much detail in the book. I tried to make it as concise as possible so that you can understand the basic concept. Now, I am trying to sum up something which you have learned in last couple of classes. And here, what I have done is that I have tried to summarize the ATP production. So you recall in glycolysis, what has happened? There are two steps where ATP is being consumed. Number one, when glucose is converted into glucose 6-phosphate. And number two is that when fructose 6-phosphate is converted into fructose 1,6-bisphosphate. So there are two steps where ATP is being consumed. But in the next phase, when ATP generation occurs, there are two phases where though 1 ATP is being produced, but because this cycle is operating twice, you get 2 ATP. So you get 2 ATP when two molecules of 1,3-bisphosphate glycerate is being dephosphorylated, and same way when phosphoenol pyruvate is converted into pyruvate. So these two steps you get 2 ATP. So what is the net gain of ATP here? 2 ATP. In one cycle, one glucose is giving you 2 ATP. But what happens during this process? You have also produced two molecules of NADH. And these NADH, which has been produced, so these two molecules of NADH are formed from oxidation of two molecules of glycerol 3-phosphate. So if I just consider the glycolysis, what you have done is that you have produced two molecules of ATP and two molecules of NADH. Now this NADH, if it is converted into NAD plus by mechanism, for example, for your lactate synthesis, or for example, alcohol synthesis, you will not get any additional ATP molecule. But if this NADH in presence of molecular oxygen enters inside the mitochondria, and of course, I have not talked about how it enters. You will be learning that there are two different shuttles through which this NADH can enter inside the mitochondria. So once they enter inside the mitochondria, they will be producing ATP. So now this ATP is being produced. So two molecules of GTP is being produced that in TCA cycle. So these are, so suppose these two steps and this steps. this succinyl-CoA to succinate conversion, where a GTP is being synthesized. So these steps are known as substrate level phosphorylation, where substrate has high energy and has led to production of ATP. And this is a direct ATP synthesis. So what happens? The energy which is present in one substrate molecule is being transferred to ADP and PI, and ATP is being synthesized. So these are substrate level phosphorylation. Now, inside the mitochondria, once NADH enters inside the mitochondria, they start producing ATP. And here, what happens, the two molecules of NADH formed in the glycolysis, each yields 1.5 molecules of ATP. So what you get? You get 3 molecules of ATP here, then 2 molecules of NADH formed from oxidative decarboxylation of pyruvate. It gives the 5 molecules of your ATP, each one 2.5 molecule. Let me tell you that this kind of a calculation is present in Israel. Earlier, each step was supposed to tell you that there are 3 molecules of ATP being produced from NADH. Now, with recalculation and calculating how many protons are being pumped out, the fraction number of ATP has been given. So each molecule of each molecule of NADH is giving you 2.5 molecule of ATP. You might be wondering why we are calculating 1.5 molecule here. Here we are assuming that NADH, when enters through glycerol 3-phosphate shuttle, some energy is required for this shuttle. And that is why the number of ATP being produced here from the NADH, which was present outside the mitochondria, is less compared to the one NADH, which is produced inside the mitochondria. This is an interesting concept. Have you understood this point? What I'm saying is that if NADH is present outside the mitochondria and it has to enter, so there is some energy involved here. And that is why once one NADH enters, it is capable of producing only 1.5 molecule. One molecule of ATP is being consumed in the transport process in shuttle, which is a glycerol 3-phosphate shuttle. If I assume another shuttle, that is the calculation is different. So here, assuming glycerol 3-phosphate shuttle, what we are calculating is that we are producing only 3 molecules of ATP starting from 2 molecules of NADH. If NADH is present inside the mitochondria, it is going to produce 2.5 molecules of ATP. And if this FADH2, it is producing lesser energy because the entry of electron is from one step below the in the electron transport chain. That is why it is producing only 1.5 molecule of ATP. So if I calculate all the steps and is assuming 6 molecule of NADH which is being produced in TCA cycle, because each TCA cycle will operate twice. Can you recollect this? Because there is a 2 pyruvate molecule, so there will be 2 acetyl-CoA, and then whatever the number. So if there are 3 steps, what we are calculating? 6 molecules of NADH, because each step is producing 3 NADH. Same way, each step is producing 1 FADH, so it is 2 FADH2 has been taken for calculation. So ultimately what you get is that each glucose, if it is converted and its electron is finally going to molecular oxygen in oxidative phosphate pathway, what you get is you get 30 molecules of ATP. So look at the conversion. If I consider just fermentation, you have got only 2 ATP. If I consider the oxidative metabolism, oxidative phosphorylation, you are getting 30 molecules of ATP. 50 times higher than what you could have achieved through fermentation. And that is why any fermentation, if I compare with the aerobic organism can create much more mass because a lot of energy is there. And the last part of this is that electron transport chain. How do you understand that there are so many steps? All these can be done by inhibitors. You have inhibitor, which can block here. So this can block the transfer of electron from NADHQ oxidoreductase to coenzyme Q. The second one blocks, which is antimycin, blocks from QH2 to cytochrome C1. And the last one is a deadly poisonous because this is the one which blocks the ultimate step of the electron transfer, and it is cyanide, azide. So these are the molecules which can block the terminal electron acceptor to accept electron from cytosol. So they are inhibitor of molecular respiration or cellular respiration. With this, I'm through with the citric acid cycle as well as electron transport mechanism and your ATP synthesis. Okay, so anything you want to ask? Any step, any question?
Excuse me, sir. Sir, I have a question regarding quiz.
Yeah, what is that?
Sir, by mistake, I uploaded my quiz on the group assignment
See,
link.
don't worry. I will be trying to check all those things which has been sent. Anything which has been sent by given time will be evaluated, and you will be getting the marks. And
No, sir.
so don't worry. In case there is you don't find uploaded